Light, waterproof, resistant, tailor-made products at a low cost. Plastic is irreplaceable, a perfect material at the center of our lives. But it becomes tricky when we need to throw it away. The world is drowning in plastic, and the artist Mandy Barker collects it on the beaches and in the seas throughout the world and displays it to illustrate what we cannot otherwise see. That plastic is now an integral part of our oceans, like algae and plankton. But paradoxically, in our seas and our oceans far from sight, we can only find a small amount of the plastic waste that we produce. So what happens to it? Is it at the bottom of the ocean? Has it made its way into the food chain? Has it disintegrated in such tiny pieces that we lose all trace of it? Scientists from around the world are on the case and are wondering whether it is modifying the ecosystem without us being able to measure the consequences. In Massachusetts, in the USA, Cara Lavender Law leads the Sea Education Association programs, which for 35 years have taught students about navigation and oceanography. Their 2 boats have trawled through the Atlantic and Pacific Oceans for years. Cara Lavender Law, therefore, has the most extensive series of samples of ocean plastic going back in time. Looking at her collection, she came up with an original question.
Because we have such a long time series in the North Atlantic, we were able to ask the question, in the region of the ocean where the plastics accumulate, is that amount of plastic increasing over time?
But what data do we have today? According to recent studies, the number of bits of plastic on the surface could be up to 50,000 billion pieces. They're everywhere, from the Arctic to the Antarctic via the tropics. They are often almost invisible because the immense majority of these pieces measure less than 5 millimeters. We also know that a good half of these pieces accumulate at the heart of whirlpools of water formed by the marine currents. Oceanic gyre. Today, we can find 5 zones of accumulation, 2 in the Pacific, 2 in the Atlantic, and 1 in the Indian Ocean. We often refer to them as plastic continents. Until now, we thought that the amount of plastic had increased over time in these zones of accumulation. But faced with her unique collection, Cara came up with another idea.
But what was interesting is when we looked at the total amount of plastic collecting in the subtropical gyre, we found that the amount is level, it's steady. We didn't see a very strong increase like we expected. We don't think that the reason is that we've gotten better at recycling or better at keeping our plastic out of the ocean, but instead we think that once the plastic is floating out here for years, somehow it gets removed from the sea surface.
Intrigued, Cara Lavender Law decided then to try to work out the amount of plastic on the surface of the ocean. Her latest study, the most thorough of our time, found up to 236,000 tons.
But this is only 1% of the amount of waste estimated to enter the ocean in a single year alone. So that's a very small number. It's surprising because there's so much plastic going in and we have no idea where it is.
1%. We can only find 1% of the plastic in the ocean. To better understand it, we have to look at the scale of plastic production. In 1950, it was 1.5 million tons per year. Today, it is closer to 300 million tons per year. The quantity of plastic floating on the surface of the ocean estimated by Carol Law is nothing compared to what we should be finding.
The danger is that people think, oh, well, it's just gone. It's not a problem because it's not there. When in fact, actually, it's the fear of the unknown. We don't know, if we don't know where it is, we don't know what it's doing. We don't know if it's having other impacts that we haven't even conceived of yet.
Finding out where the plastic is has become a major concern in order to understand its effects and its role in the ecosystem. But we must begin at the very start. How much plastic enters our oceans? Although this question has been asked since the 1970s, the first response was given in 2015 by Jenna Jambeck, an engineer specialized in the management of waste. Her study took 3.5 years.
We looked at 192 countries in the world with a coastline and a 50-kilometer buffer where waste might make it into the ocean by either being washed or blown into the ocean. And then from there, we estimated the percent of plastic that is mismanaged. We quantified and estimated that 8 million metric tons of plastic entered the ocean in 2010.
Out of the 275 million tons of plastic waste, 32 million tons are not properly managed, namely either buried, burned, or recycled. And out of these 32 million tons, 8 million finish in the water. The merit of this study is to give an idea of the scale of the problem and to show that it can be solved on land by a better management of plastic waste. According to Jenna Jambeck, if we do nothing, 10 times more plastic will enter our oceans in 2025. Once in the sea, it is too late. It's almost impossible to retrieve, and we lose its trace. However, the hunt for its trace is not new. It started around 20 years ago. One of those involved is François Galgani. He is not only well known for his studies, but also for his commitment. Thanks to him, plastic has become an indicator when measuring water quality in the European Union. In 1992, François Galgani created a groundbreaking project, checking the seabed for plastic on board Nautile, Ifremer's legendary submarine. His pictures spread around the world and were the inspiration for his model.
Donc là, on est à 20 km au large, en Méditerranée française, et 1000 mètres de fond. Et donc, c'est une plongée où on espérait voir des belles choses. Et puis, on est arrivé à un amas de bouteilles, simplement. C'est des bouteilles qui dataient des années 60 quand même. Donc on sait comme ça qu'elles peuvent durer pendant plusieurs dizaines d'années sur le fond et très profond. Et une des raisons, c'est qu'en fait, il y a beaucoup moins d'oxygène et pas de lumière du tout. Et donc ce sont les éléments qui normalement favorisent la dégradation. Et dans la réalité, en profondeur, ils se dégradent beaucoup plus lentement qu'ils ne se dégraderaient en surface, quoi. Pour l'anecdote, actuellement, les pilotes de sous-marins, lorsqu'ils sont sur le fond, ils savent qu'ils sont sur le fond parce qu'il y a des plastiques. Donc ça permet, c'est un indicateur, si vous voulez, de la zone dans laquelle on est dans un canyon. Ce qui se passe, c'est que le canyon fonctionne comme un conduit. Évidemment, tous les déchets ont tendance à descendre. Et dans les zones où il y a beaucoup moins de courant, là, ils vont s'accumuler. Et donc c'est la raison pour laquelle on trouve justement des zones d'accumulation profonde.
But these zones are difficult to access, and the data is not complete.
Il n'y a actuellement aucun bilan des quantités de déchets sur les fonds marins. Donc c'est une des grosses questions, comment dire, qui existent actuellement. Est-ce que c'est des quantités qui compléteraient justement l'information qu'on n'a pas actuellement? En tout cas, c'est sûr, si vous voulez, que ce qu'on voit, ça n'est qu'une partie, quoi.
If we don't know the quantity or the location of the plastic on the seabed, do we know more about what's happening on the surface? Are the accumulation zones really the terminus for the plastic floating on the surface? In London, at Imperial College, the oceanographer Eric van Sibyl studies marine currents. He is therefore interested in gyres, the zones where more than half of the plastic on the surface accumulates. We generally consider that these 5 gyres act as a sort of terminus, where the pieces of plastic end their journey after entering the seas. Eric van Sibyl drops buoys into the water that transmit their location and act largely like pieces of plastic. They illustrate the chaos of the oceans. 2 buoys dropped just meters from each other are found hundreds of kilometers apart after a couple of weeks. This data brings a better understanding of these accumulation zones.
Importantly, we recently found that these garbage patches are not black holes. They are not kind of like the end-all of plastic. It is not that once a plastic particle enters the garbage patch, it will stay there. Actually, the garbage patches are rather leaky. Plastic can go into a garbage patch and then slowly leak out and enter into another garbage patch. So all the garbage patches are actually connected to each other, and there's constant transport of plastic between the different garbage patches. Now we understand that actually, because there's all this movement of plastic around, everybody's plastic in the end can end up everywhere.
Eric produced a model of the route of plastic waste. Sometimes, depending on where they enter the water, it takes 10 years to reach their first gyre. Years during which they will decay. Therefore, the immense majority of pieces of plastic found are smaller than 5 millimeters and are known as microplastics. And far from what is usually assumed, these zones look nothing like the plastic continents that are often talked about.
In this room where we're sitting on now, if this would be the garbage patch, we would maybe have 30 or 40 tiny, tiny pieces of plastic. The garbage patches are a very thin soup of very small pieces of plastic. They are not islands of trash, they are not continents, nothing like that.
These microplastics have drawn the attention of many scientists. They are at the heart of the problem because they are much more numerous than macroplastics, and their ability to enter the environment seems infinite.
And what we're doing is trying to understand how different types of plastic materials break down in the marine environment. So they're in the ocean exposed to sunlight and the turbulence of waves and currents, and they somehow have gone from a large item to microplastics. And we don't know how long that takes, and we don't know how different that is for certain types of plastic versus other types of plastic. So we know that 2 common plastics float in seawater. Those are polyethylene and polypropylene. Polyethylene is used to make things like milk jugs or plastic bags. Polypropylene is used for things like bottle caps, straws, and dairy containers. So these are products that we use every day.
Cara Lavender Law looks at how pieces of polyethylene and polypropylene fragment, and which are more resistant or more fragile. The plastic splits into such small pieces that they go through the mesh of the nets used by the scientists to take their samples. Even though these holes are just 1/3 of a millimeter wide. This decay explains in part our difficulty to find the missing plastic. Richard Thompson was the first person to bring the strange disappearance of plastic to our attention in a paper published in 2004 in the journal Science entitled Lost at Sea.
We were showing now pieces of plastic 20 microns or so in size, pieces smaller than the diameter of a human hair. I mean, nobody had counted pieces that small before. So before we started looking at that, those really small pieces had not been part of monitoring. They'd not been recorded. And we were finding them everywhere we looked. So it occurred to me that this might actually represent a substantial part of the marine debris, the plastic debris now. We must keep in mind that the quantity of microplastic over the next few years will still increase because of the fragmentation of what I would call the legacy items, the large items of plastic debris that are already in the ocean. So we need to understand more about them. We need to know where they are, where they're most abundant.
Today, these microplastics are at the heart of the paradox of this disappearance. They are everywhere and yet extremely difficult to find. However, in 2014, a possible lead appeared. An American scientist, Rachel Aubard, was working on ice caps. She noticed minuscule colored fragments in her samples. She sent them to Richard Thompson.
So amongst the various different sinks, the places where substantial quantities of microplastic are accumulating, the data from Rachel suggests that the Arctic could be an important sink for particles. There's a phenomena of the formation of sea ice that actually causes particulates in the seawater to become concentrated in the ice itself as it forms. And this may be why we found such high concentrations of microplastic in the sea ice from those samples.
Extrapolating from data from a variety of samples, Rachel Aubard and Richard Thompson concluded that melting the ice cap would free 1,000 billion particles of plastics into the water in the next 10 years, constituting one of the biggest reservoirs of plastic waste on the planet. These microplastics seem to be in permanent transit. But is it possible that they lie on the seabed? No proof of this had been found until a biologist contacted Richard in 2015.
So I'm here at the Natural History Museum in London to meet Dr. Lucy Woodall. And Lucy contacted me to say that amongst some of the samples of sediment she'd been examining from the deep sea, pieces of sediment thousands of meters beneath the sea surface, she'd been finding small, brightly colored pieces. They looked a little bit unusual to her.
Oh, good morning.
Hi, Lucy.
Welcome.
Good to meet you. Oh,
Yes.
thank you very much.
Lucy Woodall studies organisms living on seamounts and the bottom of the Indian Ocean at depths between 300 and 3,000 meters.
There are coral specimens. I was surprised initially because on the cruise, my aim wasn't to find microplastics. To be honest, I didn't know they existed while I was at sea. But once I came back to the lab and I was having a look for the very small worms, the nematodes, then I was finding these fibrous items in the sediment, didn't know what they were because they hadn't previously been recorded in the deep sea. And you can see our fibres entangled right around these polyps.
In the museum's collection, Lucy shows Richard species from the depths, including worms and corals, upon which she also found microplastics.
Collected the sediment that we found the microplastic in.
Yeah, absolutely.
Not only had we never found them on the seabed, but the concentration of microplastics was 1,000 times higher than on the surface. For Richard Thompson, this is a major discovery.
So roughly what is the area that we're seeing here? You know, is it 3 or 4
It brings
meters?
the beginning of an answer to the disappearance of plastic waste. Most of all, it shows that microplastics travel through water columns to reach the seabed and therefore multiply the risks of encountering marine fauna.
If we're thinking of a risk assessment for marine life, it's the probability of encounter coupled with the severity of that encounter. So if we know where the plastic's accumulating most of all, we know where the greatest potential for risk might be.
So these are the big questions hidden behind the quest to find microplastics. How do they interact with the ecosystem? Are they totally passive, or do they affect the equilibrium? In what measure have they entered marine organisms, and therefore the food chain?
What are the possible routes, do you think, the pathways that might lead to encounter with the microplastic in these organisms?
Yeah, that's the question, isn't it? Would those microplastics accumulate? Does it take a long time for them to go through their bodies?
Yeah.
So some really great questions, but I think at the moment that's actually really hard to answer.
And these questions are being asked around the world. Not a month goes by without an article describing another affected species. In 2015, scientists had identified 560 species which had either ingested or been trapped by plastic. The number had doubled in 20 years. The large majority of organisms appear to be affected, from prey to predators, from immense whales to minuscule plankton, the very bottom of the food chain. On the shores of the Côte d'Azur, the boat from the Oceanography Laboratory in Villefranche-sur-Mer drags a manta net. On board, Gabi Gorski and Maria Luisa Pedrotti are gathering new samples. In 2014, Gabi and Maria Luisa participated in the Terra Mediterranee expedition, which took to the seas for 7 months. They found plastic all the 350 times that they cast their nets. Above all, they focused on the relation between the quantity of plastic and the quantity of plankton. Although it fluctuates in function of the locations and times of the year, the ratio can be worrying.
Il y a les petits microphyllés, c'est les petits poissons lanternes qui sont là. Il y a des salpes.
Et puis il y a du plastique.
Et les plastiques, voilà, les plastiques. Il y a certains endroits où il y a un plastique pour 2 planctons. Ça veut dire que c'est quand même pas mal. À d'autres endroits où il y en a moins. Mais imaginez-vous les petites baleines qui arrivent et qui trouvent que des plastiques à manger. Voilà, c'est ça le problème. C'est qu'au moment qu'il y a tant de plastique en mer, mais il n'y aura plus rien à manger pour les filtreurs, pour les poissons, pour les baleines.
Le problème est, je définis ça pour mes étudiants de la façon suivante. C'est pas la peine de jeter les sacs de plastique. Vous les assaisonnez bien, vous les mangez directement. Parce qu'à un moment donné, ça va revenir dans votre assiette sous une forme ou sous une autre.
This image explains the situation well and shows us the magnitude of the infiltration of plastic in the food chain. In the laboratory, they scan each sample, separating plastic from plankton. For each piece, they create a descriptive sheet with the type, nature, and form. Plastic is considered as an integral part of the marine ecosystem. It is treated and described like any other natural element that a biologist studies. A new element, which goes from level to level up the food chain.
Le zooplancton, donc les brouteurs, montent la nuit pour se nourrir à la surface. Et c'est là où les rencontres se font. L'influence du zooplancton sur le plastique intervient. Et puisque le matin, tout ce beau monde descend, c'est une oscillation comme un poumon artificiel, un poumon en fer. Ça monte, ça descend avec le plastique qui était ingéré et qui est relâché ensuite en profondeur et peut-être séquestré, peut-être dissoute ensuite. Mais c'est pourquoi peut-être il y a moins de plastique que prévu à la surface.
The microplastic interferes with the food chain and is passed between organisms. With this in mind, what are the consequences when an organism ingests a piece of plastic? Does it simply pass through its body before being rejected without any unwanted effects? Or are the repercussions more harmful than that? In Toronto, the ecotoxicologist Chelsea Rochman has been working on the subject for a few years. And if she's interested in plastic, it's because it's not so neutral once it enters the water.
In the ocean, let's imagine this is the ocean, there's lots of chemicals that are already there via runoff, urban runoff, agricultural runoff, etc. So let's pretend that all these chemicals are different types, or all these colors are different types of chemicals. And then we add our plastic. So sometimes we refer to plastics as like a cocktail of contaminants or chemicals. And this is because this plastic already has some chemicals on it from manufacturing, but you put it in the water and it's like a magnet for some of these other chemicals. So once it goes in the water and it's floating around, it's actually going to pick up and accumulate these chemicals on its surface and sometimes even within the plastics. And so then we end up with this plastic that is like this cocktail of chemicals. And then if an animal eats it, some of these chemicals may indeed transfer to the animal.
Plastic doesn't only aggregate the chemical pollutants contained in the surrounding water, it also has its own. Each plastic is unique due to the additives that we use to give it its characteristics, whether that's its color, its flexibility, or its fire resistance.
When we've gone into the ocean to see what contaminants associated with plastic can we find in an animal, the one that really seems to stand out are the flame retardants. And the flame retardants, again, are an ingredient of plastic. So some plastics by nature may already be more hazardous when they enter the water. And that's really where I think is most important for wildlife.
Chelsea Rochman is therefore looking to evaluate the toxicological effects of microplastics while respecting the environmental conditions as much as possible. She simulates the concentrations of microplastics that we find in the zones of accumulation on the surface of the oceans. She drops small plastic beads into the water so that they can fill up with surrounding pollutants before adding them to the diet of groups of fish.
And so really this right here is the liver of a fish. When we look at a fish that was fed the plastic with the chemical, this circle, this is 1/4 of the liver of this fish, and that's a full-blown tumor, so hepatocellular adenoma. And we don't expect to see a tumor like we see here in a fish that's about 8 or 9 months old, because they're really not old enough to be developing cancer and tumors naturally. So this, we think, is indeed likely caused by that combination of the plastic with those chemicals combined.
In general, scientists are not all in agreement on the question of the impact of plastic on organisms. For some, it is essential, and for others, marginal. But all agree with one point: the situation is becoming serious. The alert being sent out concerns its integration into the food chain, as if plastic was getting closer and closer to our plates. To better understand the question, Chelsea visited many fishmongers.
Thank you. For your Dorado, do those have the stomach in there as well? Is that what those are? We want the stomach. Thank you.
Take care. Have a good weekend.
Thanks.
Bye.
You too. So I'm really interested in trying to understand if there's a link between the chemicals in the plastic and the seafood that we eat. So recently I did a study and I purchased fish from fish markets in California and also in Indonesia. And we found plastic in about 25% of the fish that we purchased and in about a third of all the oysters that we purchased.
They found microplastics in the stomach of a quarter of the fish that were bought and in a third of the oysters. This data creates questions about the impact of microplastics on our health. What are the effects? What do we know? There are still very few studies, but one of these is very telling. Colin Jansen leads the Environmental Toxicology Laboratory in Ghent. His team works on mussels. They have found plastic in every single one studied. And unlike with fish, where we don't eat the stomach, with mussels, we eat the whole thing. According to their study, someone who eats a large quantity of mussels will ingest up to 11,000 pieces of microplastic per year. Colin Jansen then focused on the concentration of associated PCB to evaluate its toxicity.
What we concluded is, for this worst case, is that it's the contribution of these PCBs associated with the microplastics are still 1,000 to 10,000 times lower than the safe value recommended by the World Health Organization. So although the microplastics might be a vector, it is a very minor vector.
The conclusions are reassuring, at least concerning the intake of pollutants into our bodies. But Colin Jansen didn't stop his research. He wanted to know what happened to the plastic when it had penetrated the organism. Does it stay in the stomach for a certain amount of time before being expelled from the body? Or does it migrate to other organs? His laboratory is one of the very few to have shown that microplastics can cross the intestinal wall and migrate towards the circulatory system in both the blood and lymphatic fluid.
We see clearly in the lumen the microplastics. And the question is now, do they move from here into the tissue? And when we look closely, we clearly see here also microplastics. So not in the lumen anymore, but taken up by the tissue.
To put it another way, it can end up in other organs without us knowing the consequences. Colin Jansen is now asking the same question concerning humans. The study was done in a laboratory. He exposed human intestinal cells to very high concentrations of plastic particles measuring just 2 microns. His goal? To obtain a theoretical rate of transfer to determine the number of particles that stay within our bodies.
If you're a really big mussel eater, you will have 60 microplastics per year translocated from your seafood to your underlying tissues in your intestine. That's the value. And as I said, the main question is now, does it matter? Do these 60 particles in the worst case cause adverse effects in man? And as I said, this is something we haven't solved yet, and this is something that medical doctors need to look at and try to provide answers for.
The circle is closed. We throw our plastic away, and it ends up in our bodies. Its toxicity is being studied, and it has totally penetrated the marine environment. To a point where scientists consider today that the toxicological risks are minor when compared to ecological risks. Can plastic in its microscopic form put the entire marine ecosystem in danger?
C'est impressionnant. C'est impressionnant le nombre d'algues attachées à ces plastiques. Toutes les études sont importantes, l'étude sur la toxicité, sur l'ingestion des plastiques, mais l'idée d'imaginer que 250 milliards de nouveaux substrats se baladent en Méditerranée et qu'offrent ce pouvoir de dispersion des espèces, moi je pense que c'est quelque chose de très alarmant.
Maria Luisa Pedrotti found that 40% of their samples of plastic are colonized by algae or crustaceans. Are microplastics a major vector in the dispersion of these species? There are reasons to think this is the case. For a few years, oceanographers have considered that plastic debris are a new mode of transport. The arrival of 120 species on the Canadian coast from a large piece of plastic from the Japanese tsunami was enough to persuade them. But unlike boats, which are often vectors in the introduction of species, plastic doesn't go from A to B, but with the ebb and flow of the tides. Their ability to disperse species is thus infinite. Their slow-moving speed allows the fauna and flora to adapt. They act like rafts conveying potentially dangerous species.
Et pour moi, c'est une bombe à retardement. On a vraiment besoin de connaître quelle est l'importance de cet impact sur la biodiversité et même sur le transfert des pathogènes.
Impact of biodiversity, transfer of pathogens, microplastics are raising questions. And studies tackling these questions began at the Marine Biological Laboratory in Woods Hole. Since 1888, this laboratory, decorated with 56 Nobel Prizes, explores fundamental biology and biodiversity. Linda Amaral Zettler, a microbiologist, has included plastic in her research interests.
So here we have a liter of seawater. And believe it or not, there's about 1 billion bacteria in this one sample here, and 10 to the 4th smaller organisms, also microbes, but that are sort of like the plants that live in the ocean that give us about half of the oxygen that we breathe. And to it, I'm going to add one of these pellets. So just by adding this one drop of plastic, we're transforming this community into something we don't know yet. And that's because these kinds of surfaces don't exist naturally. So this is part of this new habitat that we're trying to better understand.
In 2013, Linda Amaral Zettler and her husband Eric focused on organisms living on plastic debris collected from the ocean. A new world opened up in front of them. A unique world. They called it the Plastisphere.
Hi, Eric. How's it going?
Good.
What have you got there?
So I've got both some of our incubation samples. So here's the edge of the plastic. There's actually some animals, some invertebrates. This is a hydroid right there, and then it's a colonial animal, so those are coming off there. And you can see the surface just looks kind of crusty, but if I start zooming in, you can see that it's just loaded with diatoms as we zoom in. The whole surface is practically covered with these pennate diatoms. You can see how many diatoms there are on this one, but there's all kinds of stuff.
Yeah, no, it's covered.
I don't know what that is, but I want to look at it.
Okay. Oh, what is that?
I have no idea.
At first I thought it was a ciliate lined with bacteria, but it's not.
Yeah.
It's something else. Oh, I don't know.
Oh my God.
It is the ciliate.
Wow.
It's the ciliate, no?
Yes. But what's interesting about this particular one is the surface is completely covered with little bacteria. See, each of these is a bacterium. You can see some are dividing. That one's just starting to divide. That one's almost completely divided. These are dividing, so we know they're growing. They're not just stuck there.
The very first time when we looked at these pieces of plastic, now remember, these are really tiny, probably half the size of my thumb, my pinky nail. What we saw were some very fascinating, basically little worlds, if you will, of microbial life. We found predators, we found prey, we found organisms that will actually parasitize other organisms, and even symbionts. So this told us that it was really a self-sustaining little ecosystem that was floating out there in the ocean gyres and garbage patches.
They discovered a world in itself, an ecosystem that existed alongside others, special communities, different to those that developed on a natural medium such as wood or algae. And among the organisms identified on the pieces of plastic, one of them is intriguing.
In one of the first pieces of microplastics that we characterized the bacterial communities on, we found that there were very large numbers of these vibrios. And vibrios are interesting because some of them, not all of them, but some of them cause disease in animals and humans.
Vibrio bacteria constituted up to 25% of the microbial cover of a piece of plastic, and some of them are known to carry cholera. This finding requires at least some attention. Tracy Mincer, associated with Linda and Eric's research, has shown that the vibrios had specific attachment mechanisms which allow them to stick to plastic very quickly and very well. These bacteria also have genetic characteristics that allow them to attach themselves to fish's intestinal tissue, causing a real danger to fish stocks.
They don't have the really serious pathogenic abilities that would affect humans. And it makes sense because these vibrios are not interacting with humans at all. They're interacting with fish. And so what we think might be happening is that the plastic could serve as a vector for these vibrios to get around and get into other fish and transmit to a parasitic or pathogenic burden to fish stocks.
A Trojan horse for pathogenic bacteria and a raft for invasive species. These microplastics could be the vector for significant transformations in the marine environment. Studies are in their infancy, and yet scientists are already alerting us to the danger that they could be harmful for the equilibrium of this environment. But when observing these microbial communities, the three American researchers also made another surprising discovery. Some of the bacteria have the ability to accelerate the process of decay of the plastic.
A surprising finding was that many of the microbes that we were finding in the plastisphere had also been found in oil spills, for example, and other hydrocarbon degradation types of experiments. And so we found that was really compelling based on the idea that microbes that are associating with these plastics may also have the capability to degrade plastics or those hydrocarbons. We're still looking into that, but there's some good evidence by microscopy that some of these microbes are actually able to accelerate the weathering or the hydrolysis, the breakdown of plastics in the wild, in the open ocean.
Is in the scanning electron micrographs, we saw these things we call the pit formers. So these are little 2 to 4 micrometer round cells, spherical cells, that appeared to be embedded in pits in the surface of the plastic. Whether they're metabolizing or just physically breaking it down by the way they attach, we don't know, but that's one piece of evidence that they're impacting the plastic and perhaps breaking it down.
Bacteria capable of decaying plastic? Eric, Linda, and Tracy examined this question, and there is now no doubt that they have a role in the fragmentation of plastic in the oceans. But are the bacteria able to metabolize and therefore cause the plastic to totally disappear from our oceans? Today, there is evidence that this is the case.
Donc ici, on met des morceaux de plastique qui sont restés en mer pendant 2 mois. Pendant ces 2 mois, il y a eu des bactéries qui se sont développées à la surface du plastique et on va vérifier maintenant s'il y a des bactéries qui sont capables de dégrader le plastique. Alors la façon de le démontrer, en fait, c'est de mettre ce morceau de plastique avec les bactéries dans un milieu minimum où il n'y a plus que du plastique à manger. Voilà, ce qu'on voit avec ces courbes, c'est qu'on a une diminution de l'oxygène, c'est-à-dire que les bactéries sont effectivement en train de manger le plastique puisqu'elles sont en train de consommer de l'oxygène.
In Bagnols-sur-Mer, Jean-François Guiglione and Claire Dussud from the Microbial Biology Laboratory show that there are strains of bacteria that, when isolated, totally consume different types of plastic from the ocean. The bacteria have developed a complex strategy for ingesting the plastic.
Donc un plastique, en fait, c'est un polymère. Donc si je prends par exemple le polyéthylène, qui est le plastique le plus communément utilisé dans le commerce, c'est une chaîne de carbone et d'hydrogène. Donc sur cette chaîne de polymères, il n'y a que du carbone et de l'hydrogène. Et ça, c'est un problème pour les bactéries.
It's a problem for bacteria because these plastic particles are not edible and are too big. Bacteria therefore start by sending enzymes outside their cell to oxidize them. Once there is oxygen on the polymer, the bacteria send other extracellular enzymes to cut the chains of carbon. Different species of bacteria are implicated in the process of decay until they are able to reduce the polymer to small molecules that they can finally consume. Can bacteria explain why we cannot find 99% of the plastic in our oceans? Could they be the answer to the problem? Claire and Jean-François are working to characterize the microbial communities. The process of decay of plastic in the sea is complex. Tens of thousands of bacteria are implicated and follow each other over time without us knowing exactly who does what.
C'est une bonne nouvelle parce que ça veut dire que la façon dont on arrive à sélectionner parmi les espèces rares, justement, on arrive à les sélectionner, on tente actuellement de les isoler pour mieux les connaître. Donc ça, c'est quelque chose de positif parce qu'on arrive à mieux comprendre le fonctionnement. Par contre, ce qu'on se rend compte, c'est que ces bactéries, en fait, elles poussent très très lentement. Et l'utilisation, la dégradation du plastique, c'est un phénomène qui est très très lent. Donc ça, petit à petit, on arrive à comprendre aussi que dans la mer, la solution, ça ne va pas être les bactéries qui vont dégrader ces plastiques, parce qu'il y a une telle quantité de plastique qui arrive, le temps de dégradation est tellement lent que les bactéries en fait, n'arrivent pas à manger tout le plastique qui arrive en mer, ça c'est évident.
If bacteria can absorb plastic, they can still only explain a tiny part of this disappearance. They are not the miracle solution to the mystery that remains. Nature can adapt, but it cannot follow our frantic consumption and our inability to manage the resulting waste. For a few years now, the scientific community has tried to understand what happens once plastic enters the sea. Questioning not only its fate, but also its effects, they have identified microplastics as a source of preoccupation and are already wondering whether nanoplastics, even smaller particles, are not even worse. But today, we are still measuring the time and the rate of decay of these plastics in our oceans.
We're very much at the first step. We're asking the most basic questions. We're just trying to assemble all of the information that we can find in order to see if we can answer these questions. For the most part, we can't.
But on the other hand, you know, we do have some answers and some knowledge about ways that we could perhaps prevent plastic entering the ocean.
Way to keep plastic out of the ocean is to stop putting it in there. Stop it at the shore, the rivers, the landfills, the waste dumps, you know, just human choice, basically.
So it's not about not using plastics, it's about using them much more smartly. And what that, in my view, is going to take is rethinking the way we produce, use, and dispose of plastics. And the increasing realization is that there really isn't any way on the planet.
We are living in a vacuum. It is time to realize this before this piece of art that we come across by chance in an airport morphs into something more sinister, no longer there to calm the stressed out travelers, but to remind them of what has become of our oceans.